All right, it's 2 minutes after the hour. So I'll start the meeting by introducing Karla Saavedra Rodriguez. My name is Brian Foy. I don't know everybody here, but I'm taking the place of introductions for Tim. I'm a professor here at CSU. I'm one of the co-PIs of the RaHP VEC grant, along with Natalie Marzek at the CDPHE. So I know Zane because he's in our lab. I know Tatum, Maddie, I've met you. I don't think I know the people in Grand River. Well, I've probably met you all before. Lawson or Caitlin. Where are you all at?
I am in Utah.
Oh, Caitlin, I met you.
Yeah,
Okay. I
briefly.
was thinking you were somebody else on the screen. Sorry. Nice to see you again. Are you the one pulling our wings and trying to do the wing scales? Or
I
is that
think I'm gonna help out with that a little bit. I'm kind of doing a bunch of different things, just volunteering as well.
It's good to be like very diverse and volunteer for a lot of things. You get a lot of stuff. Lawson, how about you? Where are you at?
Oh, I'm just here in Fort Collins.
Oh, okay. Maybe you're down there. Okay, sorry. I've seen you. Am I missing anybody? Grand Rivers people? No. Well, anyway, nice to see you all. I hope you're all enjoying your, oh, there we are. Hello, we're from the CMU in Delta. Oh, I met you all. Great. I hope you all are enjoying your internships so far. Any questions for me about what's going on that you, or anything you want to complain about? Because I'm the one I can take your complaints. I hope Tim's taking good care of you. Okay, so today for this internship, we're going to have Dr. Karla Saavedra Rodriguez, who's a colleague of mine in the CVID and professor at MIP. She's been at CSU for a long time now, and she is an expert in insecticide resistance. And so I'm gonna actually jump off, and Karla, it's all you, and you can close this meeting when you feel like it. Okay.
Okay. So
Right.
we have like a whole 45 minutes, I think, or
yeah, I think
hour,
you could take as much time as you want. I think, well, not past the full time, I guess. And make sure you you're going to give a little bit of your background for them, right? So that they know it's, how you came up and stuff like that. One of these days I'll do one of these myself and tell you what my background is. Obviously, part of this is career development for you all. So it's important that you get some of that as you sit in these lectures and sleep. No, you won't sleep with Karla. Okay. All right, I'll take it away, Karla, and I'm going to drop off.
Wait a second. I'll just try to share my slides and just to see if that everything is okay. And I should share my screen or my presentation. Okay.
We can see it.
You can see it? Okay.
Yeah, but you're not in presentation mode. Oh, now we see your notes mode.
Okay, so let me just display. There?
Yeah, there you go.
Okay.
All right. Take care, y'all. We'll talk later.
Okay.
Good luck, Karla.
Thank you. Okay, but thanks for joining, and it's really nice to meet you. I have seen some of you before. So my name is Karla Saavedra Rodriguez. I work at Colorado State University. I'm an assistant professor, and I joined the RaHP group when we start writing this grant. And as Brian said, I've been working on insecticide resistance for 15 years now in different species of mosquitoes. So I will provide my expertise on this for this grant, and also if you have any questions or if you want to try to do some insecticide testing, just please write me an email and we can start. I can guide you and show you what I do and and how we can improve. So I think they want me to talk a little bit about my background and what I'm doing. And well, I'm from Mexico. If you feel an accent, yes, that's I speak Spanish, and I was born in Monterrey, Mexico. I just put a picture here. It's an industrial city. There's like 5 million people living in the city. They have like a very hot and humid summers and spring. And then on the winter, we can get like zeros, like centigrade. So it gets cold. And so it's very extreme. The weather is very extreme. It's a developing city. So we will have like very beautiful houses, and then you go 2 blocks and then you see very poor housing. So in these areas, I grew with dengue epidemics all the time. I I was remembering that one of my first memories about dengue, it was like in my streets there was a prosecution. There was a dead kid on hemorrhagic dengue, and it was a neighbor. So I can remember those. So I grew with the burden of these diseases in the city. So I study biology. I put up here a picture. Is the university that I went to. They have a really strong biology program, and that's where I joined an entomology department. At that time, there was like a lot of aquatic entomology and like biological control of larvae for mosquitoes. So all the group was working on this. So there was no room for me to do another experiment. So I switched. I started like digging into insecticides, right? And that's how I started working on that. Um, then I joined a master's degree program in medical entomology in the same university. So there are two things that made me like uh, really love medical entomology and vector control. And one was a workshop that I did in malariology, is malaria studies, and it was down in Tapachula, Chiapas, southern Mexico. So that's where I like fell in love with all of the different um systems, all of the different vectors, and how all of the social problems also affect the burden of these diseases. So it was, for me, it was very eye-opening, and that's where I felt like I needed to do this, right, for a living. So there's a second thing is that during that time, I'm talking 2000, maybe many of you were not born at that time, probably, right? So, so I there was this collaboration between my university in Monterrey and CSU. So they have these international training grants. Here in Colorado State University, we had like Barry Beatty, Bill Black. So they were like writing these grants, inviting people from Mexico to come to the US, get trainings on how to control mosquitoes, how to study vector competence, a lot of different things. So that's how we got like I was in the university when they started this collaboration. So I was part of those collaborations. First, well, this is the El Esposo. This is my husband. I was still studying my master's degree. I didn't know how to speak English, and so I couldn't apply to these trainings directly. But my husband did, because he's also a medical entomologist. So that's when he moved to Fort Collins to CSU, and we joined. He joined Bill Black Laboratory. He was a brilliant geneticist and vector biologist. So and then he also did statistics. So he was a very strong person and a very strong researcher. So we joined his lab here at CSU, and he was doing at that time like a lot of molecular biology techniques because it was that era. And so I started volunteering in his lab for maybe 2 or 3 years because I was on a student visa, so I couldn't work or study, or I was depending on my husband's. So after volunteering, I also took some biology of disease vector courses that they were teaching. So then we went back to Mexico because the idea was to go to learn things here, go back to Mexico and work there. But my husband never found a job there. It was like one of those periods where you cannot find jobs in a city in Mexico when they don't care about vector-borne diseases, right? So then he started a postdoc, and then I joined. I was accepted on a PhD program on medical entomology in Mexico, and Bill reached me and he told me like you should come back to Fort Collins and let's do your research here. So I came back to Fort Collins. At that time, we had this grant with Bill and Melinda Gates. They were doing a lot of insecticide resistance. So that's when I joined Bill's group. I already have the experience doing toxicology, but also because I volunteer, I had all of the molecular biology techniques that I needed for my research. So after that, he invited me to do like a postdoc. Then I switched to a research scientist, and then to an assistant professor. So it's been a long process, but well, this is mosquito biology and control is something that I find very fulfilling because you mix your interest for biology, for molecular biology, but also for different ways to control mosquitoes and help people, right? Try to get rid of these mosquitoes and the burden of the diseases they transmit. So that's just a little bit about me. During my research in CSU, we've been working a lot with the system of Aedes aegypti and dengue transmission in Mexico. So we have been doing different studies in Mexico, but also combining like the field studies with molecular biology and trying to understand, to go a little bit deeper on how this transmission is occurring, how insecticide resistance is what is happening with insecticide resistance. So, okay. So now I'm gonna switch to what we are doing here in the I put Rocky Mountain. It's Rockies and High Plains Vector Training and Evaluation Center. So I'll be working on this insecticide resistant testing. The system is very different to what I have worked before. Before it was mosquitoes, which breed in the back patios of the houses. Here we have a different insect, right? A culex, which breeds whatever he wants, right? So his distribution is larger. So we know that these culex mosquitoes feed on birds, which they're the major carriers of West Nile viruses. So eventually, some of these mosquitoes will bite a bird, they will get infected, and if by any chance they find a human or a mammal, they will feed on them, and they can transmit the disease. Luckily, like humans and equids do not they get infected, but they don't produce like large West Nile viruses infection, West Nile virus infection. So that's why they are considered like a dead end host. So they won't be able to transmit. If a new mosquito, non-infected mosquito bites a human, if an infected human, the mosquito won't get infected because the viral loads in the human or the equids are very low, right? So that's where they are considered dead end hosts. But birds is another thing. They will amplify the virus. So in different mosquitoes transmit West Nile virus. We have mostly like Culex pipiens, Culex quinquefasciatus, and Culex tarsalis. So they're really good at transmitting the disease, but also they are widespread in different areas. They prefer to feed on birds, right? But they eventually will find humans, and they will also bite other mammals. So that's why they are the major mosquitoes transmitting. See, here is just a map showing the distribution of Culex, some of the Culex species mosquitoes. We have Culex pipiens in the US, Culex tarsalis, if you see, like they're distributed everywhere, right? So that's at least for our area in the Rockies and high plains. That's why we are interested in studying Culex tarsalis. Also, these mosquitoes have a very large, they can adapt to different environments, right? So this is just a study showing how some of mosquitoes, the same mosquitoes can be in a urban or in a city, they can be in peri-urban areas, rural or wild, right? And the access to different sources of blood, it will change how they spread the disease to humans, right? So this is just something that is important to keep in mind, right? Not not all of the species will play an equal role in the transmission, and it depends where are their hosts, right? What are they feeding on? Here in Colorado, we have Culex pipiens and Culex tarsalis transmitting the disease, and as you know, like this, the mosquitoes have a life cycle, right? So most of the life, so we have eggs, larva, pupa, and then an adult. Most of their life will occur in the water, right? It's an aquatic cycle, maybe 7 to 10 days, right? And then when the adult emerges, the females will look for blood, and that's probably from birds. And then if they get a blood meal, then they will have the energy to produce eggs and lay eggs, right? So this from blood feeding to produce eggs, it will take like 3 or 4 days, right? So it's pretty fast, and they can feed several times during their lifetime. So that's how we start with one mosquito, and then you end up with thousands, right? In very few in a few months. So this is just an example of what kind of larval habitats you can find these mosquitoes. It can be like domestic, like produced by our like domestic areas. It can be like marshes or ponds and swimming pools and so on. So this is just some pictures of Culex tarsalis. If you see, like they lay these eggs on rafts, right? So probably between 50 and 100 eggs in a single raft. Then the larvae will grow in water that has a large organic material, right? And then if you see there, they have this, well, there's the head, the body, and then they have the siphon. They breathe atmospheric air. So that's when you will see their behavior, they always try to get to the surface and breathe through their siphon. Then they switch to pupae, and the pupae also need to breathe atmospheric air. If you see, they have like it looks like antenna, but these are like trumpets, and they are getting their air from those trumpets. So after this pupae, we will have the adult, females or males. Females will, well, both of them can feed on sugar, like flowers, nectar from flowers, but then the females will require a blood meal to get enough energy to produce eggs and continue the cycle. So only female adults will be important for West Nile transmission. What do they feed? That is a different story. As we were telling, like they will prefer birds. So they really like American robins and doves. This is a graph in 2007, I think, yes, in Colorado, and they collected Culex tarsalis mosquitoes, adults, and if they had a blood meal, they check what was the source of that blood, right? And if you see, like, they check in June, July, and August, and then most of them will feed in American robins, right? And a lot of them. Then we have the doves. And then as summer goes on, for example, in August, you can see an increase in human blood in these mosquitoes, right? Once, because there are more mosquitoes, maybe birds migrate or maybe birds are not available. So the blood meal composition will change over time. At least here in this area, for example, most of the cases in the US will occur between July and September. So there's some seasonality. So that's when mosquito populations are very high, and that's where birds migrate, and that's where we have a lot of people outside, right, during the summer. So that's when more infections occur. So this is important because it gives us an idea of when should we do surveillance and when should we intervene, right? When should we be ready to expect West Nile cases? So there are different ways in which counties decide where to apply mosquito control, right? So the idea is that there's no vaccine against West Nile. You cannot control how birds fly around the area. So the only way you can control the disease is by controlling your mosquitoes, getting your mosquitoes populations under a certain threshold so they cannot transmit the disease. So there are different ways in which counties decide when are we ready to do mosquito control. And one of the most used is the vector index. It's just a measure of the risk of transmission of the pathogen within a given area. So it's you collect adult mosquitoes, you get the number, you pull them, right? You get the number of West Nile positive mosquitoes and divided by the total number of mosquitoes, and then you multiply by 1,000. And then the higher the number of the vector index is, like, the more risk you have to have West Nile. And certain counties say, like, oh, we have a vector index above this number, then we can consider applying insecticides, either to kill different stages of the mosquito. So I was telling you, like, when we talk about suppressing mosquito populations, you can remove the breeding sites by not allowing eggs, egg laying, and then you can apply larvicides to kill the larvae, right, in water, and then you can apply adulticides. So there's pros and cons for each of these ways of controlling, but usually when we try to apply adulticides, it's because we have an emergency, right? And we already have West Nile infected mosquitoes, and we need to just mitigate the population fast, right? So that's these adulticides are mostly used during emergencies, while other forms of control, for example, larvicides, you can apply them just to prevent that your populations will increase by a certain threshold. So there's several insecticides. Most of them are targeting agricultural uses, right? So there's around 26 insecticides with different mode of action, and they have been discovered in different eras, right? Like we have the golden age when DDT and organophosphates were discovered in the 40s and 50s. Then we have, like, the pyrethroids and the new age of insecticides. So every time as more research is being done, they're discovering new ways of new molecules that have different modes of action. And we want variety, right, in this take. So one of the major problems with insecticides is that they're very expensive to make, right? And it takes a long time for them to be formulated and also to be accepted for certain uses, for agricultural use. But if you want, again, to use them for public health, that's another set of requirements that you need for your insecticides. So it might take 2 or 3 more years to get your formulation. So it's an industry, right? And there's many factors that we have to consider when choosing these insecticides. So this is a paper where they talk about the vector control insecticide use. There are major insecticides classes that are used for vector control. We have the pyrethroids, like the organophosphates. We have orgachlorines like DDT, and carbamates, right? So these graphs just show, like for example, this is in Africa. In blue, they're using pyrethroids only for bed nets. So that's to protect the mosquito that transmits malaria. They like to bite during the night when people is sleeping. So a way of controlling them is by using a bed net, right? So these bed nets are treated with pyrethroids, and then when the mosquito tries to bite a person in the night, it gets in contact with the bed net and it's killed, right? So if you see like the large amount of insecticides used just for bed nets, and it's pyrethroids, and there this in figure B, we have like out of Africa, mostly Latin America and Asia. This is the vector control activities still use a lot of pyrethroids, and DDT, and then lower amounts of organophosphates. So this is just to show you that, and this is still the case, most of the insecticides used in mosquito control belong to this class of pyrethroids. Then for larvicides, there's like a more variation, and many of these insecticides are based on bioinsecticides. So for example, we have these larvicides are applied by different ways. Backpack applications, we have trucks, helicopters, or drones, right? So there's different ways to apply these larvicides. And the most important, at least here in the US, are the bacterial larvicides. We have insect growth regulators and oils and films. So these bacterial larvicides, maybe you've seen them. You can go to Home Depot and find these mosquito dunks. This is BTI. BTI is a bacteria called Bacillus thuringiensis. The bacteria produces a toxin, right? So they formulate this insecticide in a way that the larvae feeds on these dunks, right? It feeds on the bacteria, and then inside the gut of the larvae, the bacteria activates, it creates this toxin, right? And the toxin will bind to the intestine and make pores. So it breaks the midgut. So the mosquito dies because he cannot feed anymore. So that's a way of, these insecticides are very specific for certain larvae. Some of them work only for Culex pipiens, while others will work with Aedes aegypti, for example. So this is one of the insecticides that are very specific to larvae. There's also other kinds of insecticides, such as these insect growth regulators. They're based on research that was done for like trying to understand how mosquitoes go from larvae to pupae to adult. There's different hormones produced during their molting, right, process. So they discovered that if you create like a juvenile hormone analog, you will prevent the mosquito from metamorphosizing, right, to change to the next step. So that's when the larvae is forever young, right? It gets along forever, and eventually they die because they cannot get into the adult stage. And then another larvicide that is widely used for Culex mosquito control is oils and films. As I was telling you, there's like the larvae and pupae feed on atmospheric air. So if you look at their siphon, at the end of the siphon, they will have a lot of hairs, which create this, they break down the surface tension with the water, and they allow the siphon to connect directly to atmospheric air, and that's how they breathe. So when they apply oils or films, this tension, the surface tension is broken, and then the mosquito cannot breathe anymore, and they suffocate, right? The problem is that if you add this to to a lake or to a large pond, you not will only kill larvae, but other insects that also breathe the same way. So you have to take into account environmental damage that you can do by applying these insecticides. Then I'm going to switch to the adulticides. So there's, I show you like in the other graph, there was like 26 different modes of action, but really for mosquito control, we just have two, right? That are approved by different, like for the Environmental Protection Agency or by WHO, the World Health Organization. So we have organophosphates and pyrethrin or pyrethroids. So organophosphates are all there. They are not very specific for insects, right? So organophosphates are also toxic for humans and mammals. That's what I'm trying to say. While pyrethroids are more specific for the insects. So these insecticides are usually formulated in a way that they can be sprayed in very small particles, right? So the idea is that when you spray small droplets of insecticide to the air, they eventually will make contact with a flying mosquito, right, or a resting mosquito. That insecticide will make contact and will kill the mosquito. So there's to apply this kind of control, you need to know when are mosquitoes active, right? For example, for Aedes aegypti mosquitoes, the ones that transmit dengue, they bite during the morning and they are active during the day. So you have to apply that insecticide during the day. But if you go to Culex, right, they will start biting at 7, 9 p.m., right? So that's why they have to apply these insecticides when the mosquito is active at 9 p.m. during the night. So you have to take into account different, the behavior of the mosquitoes and also like the characteristics of each insecticide for the formulation. So these insecticides, I'm telling like they have a different mode of action. Pyrethroids are sodium channel modulators. Organophosphates are acetylcholinesterase inhibitors. So both are considered neurotoxins. They act on the neural system of the mosquito. And they have, for example, these pyrethroids, there are 16 registered molecules or formulations approved for use in the US, while we have more organophosphates registered for use. However, these organophosphates also like they have a smell. People don't like them, so that's why most of the people go to pyrethroids. Pyrethroids act very fast, right? So they kill a mosquito within minutes, and organophosphates take longer, like maybe 24 hours. So all of these things help make decisions about what you will use for mosquito control. Another important thing is that there's in pyrethroids, there's like a high cross resistance. That means that, for example, you have different molecules. If your mosquito population is resistant to permethrin, it's very likely that it will be also resistant to deltametrin, right? So that's called a cross resistance. So getting resistance to one, you get resistance to everything else. So that limits the insecticides you can use for control. This is not very common in organophosphate resistance. So then okay. So we have the ideas. I mean, what I want you to get home is like there's very few molecules approved for public health, right? One, because they are very heavily regulated by EPA or by WHO. Also, we have very few intervention types. The way you can apply insecticides is limited to one or two, right? And from all of the insecticide industry, public health is just like 10% of the interest, right? Most of the research and most of the formulations go to agricultural uses. So that make us have less and less molecules for public health. So because we just have like 2 or 3 insecticides, then that means that when you do control, you have to overuse them, right? So you end up using the same insecticide for 10 years or 20 years. So that's generating a lot of selection pressure, right? On your mosquito populations. And then if you have a program where you're not monitoring what's happening with your mosquitoes, then you will end up at some point with insecticide resistance in your populations. And this has been seen not only in mosquitoes, in all agricultural pests. So once an insecticide is released and used like for long terms, then you eventually will see resistance. So until now, there's like this report, 14,000 cases of insecticide resistance. They evaluated, this is a report of 300 insecticides and 602 species. So insecticide resistance will happen. So we need to be smart how we use them, right? So here we have just like a selection of insecticide resistance model. Well, just I'll stop here a little bit. And do you have any questions or comments or something that you want to share at this point? No? Okay. If not, let me know. I can stop at any moment. So we have a selection of insecticide resistance. Usually you have your population of mosquitoes. There's going to be 1 or 2 mosquitoes that will be different, right? Everything starts with when someone is different. If by any chance that difference is if you apply an insecticide and that difference allows the mosquito to survive, then you will see that the next generation you will have more mosquitoes with those traits, right? With insecticide resistance. If you apply the insecticide again, that mosquito will leave more offspring because it's not dying while the other ones are dying, right? The susceptibles die, your resistant gets alive, lays eggs, and every generation, if you keep applying the same insecticide, the frequency of these resistant mosquitoes will increase over time until you end up with when most of your population carry these resistance mechanisms, right? So we never want to get into this point, like 0.4, where we have like most of them resistant, because then it's very difficult to go back to susceptibility. So how mosquitoes get resistant is a different story. You have to look at each mosquito individually. There are different mechanisms. Some of the mosquitoes have like avoidance behavior. They don't make contact with insecticide anymore, for example. They know how to sense it. Sometimes there's like cuticle modification. So the cuticle is the exoskeleton of the skin of the insects. So sometimes they get thicker, so the insecticide doesn't penetrate that easy. So that's a mechanism of resistance that has been found in mosquitoes as well. Some mosquitoes are some of the mechanisms is like developing a very strong detoxification set of enzymes. So when the mosquito or the insecticide gets in the mosquito, these enzymes break down the insecticide pretty fast, so they are not get killed. Also, sometimes some mosquitoes, there's a target site, like for example, when I was telling you the sodium channel modulators of the acetylcholinesterase, that's the target site of these insecticides. And some mosquitoes, they develop mutations, right, where these mutations don't allow the binding of the insecticide, and insecticide is no longer effective. So these mechanisms can occur one at a time, or they can be several in the same mosquito, right? So, but they're usually developed or selected just to protect against one insecticide at a time. So when we want to study insecticide resistance, there's different way we can study. The most important is like toxicological bioassays when you actually expose the mosquito to an insecticide and try to see, okay, it dies or not dies, and what's the proportion of the population dying with this insecticide. Also, there's like biochemical assays where you ground the mosquito and then check if they have a particular group of enzymes, for example, esterases, and you say, oh, these esterases are really good at breaking down insecticide, and this population is overexpressing these enzymes, right? So you can infer that they are resistant because of this mechanism. And also there's like molecular assays where you can look at the target sites of each in each mosquito and try to find the mutations, if they have the mutation or not. So here is toxicological bioassays at the operational level is the best way to check for resistance, right? Because you have your population and you can see what happens if you expose it. There is different bioassays to test for resistance. In the US, the CDC bottle bioassay is very popular. We have larval bioassay, we have the WHO cylinder assays, and another the WH cone assay. So in most of them, you use mosquitoes, you expose them to a surface impregnated with the insecticide. In this case, the bottles are coated with the insecticide inside, and in the larvae, you have to put the insecticide in the water and so on. So there's different ways to analyze, and there's like some of them like to expose them for 30 minutes, other for 1 hour, other to so there's a lot of variation in how you measure a resistance. And the idea is, why do we want to monitor insecticide resistance? One, we need to know which regions have or which populations have resistant, and also we need to know like what insecticides we can use to control our mosquito populations, right? So that's at the operational level, that's the most important point. So in this case, we in the Rockies and High Plains VEC, we want to do this insecticide resistance monitoring in Culex mosquitoes, because we are in an area where Culex tarsalis is very it's well dispersed. So we want to focus on Culex tarsalis. But if we end up finding more Culex pipiens, then we'll do Culex pipiens as well. So the idea of this resistance monitoring is to generate enough data to know like how mosquitoes respond to different insecticides, and also help like the different vector control programs to make decisions about what insecticides they can use because they could be effective, right? So we want to help them and to make those kind of decisions. And the idea is to collect mosquitoes from different areas in the Rockies and High Plains, and then if they can send them to CSU, or if they can do their own bioassays, right? It can be adulticide or larvicide, to generate this data and then share these results, build a map, communicate these results to the at the local level. So that would be the main goal of having this insecticide resistant monitoring. So there is, I don't know if you have heard about the vector serve, right? So in them, they report like West Nile cases, but there's also like a tab for insecticide resistance, and they're like having a lot of data. They do the bottle bioassays, and then they upload the data, and they can show like, oh, these mosquitoes from California in this area, like they have 90% of them die, or 50% of them die, or they have low resistance or high resistance. So that's the main idea. If you check, there's a lot of data on California. There's a lot of data for the East Coast, right? But for the middle, for Colorado, there's like very few, like less than my fingers, studies on insecticide resistance. So that's where we want to, everything that we will generate doing this study is going to be important to inform about what's the insecticide susceptibility or resistance. So here, this is like a commercial, and I'm just RaHP, we're doing this. If you need help determining your insecticide resistance status, we're doing at CSU, we'll do the testing, or if you want to do them in wherever you are, like you can also, like we can help you develop all of the system to do the bioassays in situ. So the idea is, it says like only 30% of mosquito control districts in a region know the insecticide resistance status of their local mosquito vectors. So it means that, yeah, 60% of them are not doing any insecticide resistant. They're not checking for insecticide resistance. And with those very few that have done, most of them find insecticide resistance in Culex mosquitoes. So what we want to do is to help in this process, right? To test more mosquitoes. So we're doing this submission program. Team did this. It says free IR testing, and we include a free sample collection kits. So the idea is to ask you, like interns can help us in this also, to collect mosquitoes and send them here. Or if you, yeah, or if you want to, if you prefer to do them where you are, we also can help you to have everything that you need. So the idea is, we ask you to collect larvae or rafts or adults, whatever you are capable of collecting in your area. If possible, send them to CSU, and then here we can do the bioassays. So one of the things that gets a little bit complicated for Culex tarsalis is that not a lot of people is able to find egg rafts, right? If you go to different areas where the rafts can be, it's hard to find them. So, but it's not impossible, right? If maybe you know that specific spot where you can find always larvae or adults. So there's different, well, the first step is to identify these larval breeding sites. You can use these dippers to collect larvae. And you dip, there's different techniques on how you can dip. So here's just a list of the different techniques you can use. And also I put the link for this YouTube movie where they show how to do each one. So it's, I thought it was just like as simple as dipping, but not the way you dip is, it can affect which species you collect. So it's, if you planning to do this, I recommend watching that that video. So the idea is to have around 150 larvae to test one insecticide. If you want to check for BTI larvicide, we need 150 larvae. So it's usually 4 replicates and 1 control. The 4 replicates is, each one has 25 mosquitoes in each cup or in each bottle. And the control, you don't use any insecticide. So, so let me show you here. It's just like you dip, here they are dipping egg raft. They putting them in falcon tubes, and then they chip them, right? So we are providing, if you're interested, we can provide these chipping kits, right? They have like tubes, they have the dippers, they have plastic bags, so you can send us either rafts or larvae. So this is just an idea how you can chip the larvae. If sometimes if you cannot find adults, we also can accept like adults, right? So this is the CDC mini light trap. Um, usually for culex, you set up these traps overnight. The mosquitoes, oh, it doesn't look, you cannot see here, but over the top, there should be like CO2 cooler. Freon is releasing some CO2, so that will attract the mosquitoes. Usually this will be mosquitoes that want to blood feed, right? Because they're looking for CO2. And then there's this fan, so the mosquito will get sucked by the fan and then get into this mesh container. So if you do that overnight, and we're lucky, you can get 150 mosquitoes, then the next day we can transfer those mosquitoes without killing them, put them in this like bug dorms, and then the next day we can do the bottle bioassay from field mosquitoes. So it's if you don't find larvae, we can find adults, and we can try to test whatever we collect. So the bottle bioassay is very simple, right? You have to prepare your bottles with different insecticide doses, or diagnostic doses that are recommended by the CDC. Here is like in micrograms per bottle. You coat the inside of each bottle with acetone. Acetone evaporates, the insecticides stick into the inside of the bottle, right? So then you have to put your mosquitoes inside the bottle and count like how many die every 5 minutes until a diagnostic time. And here the CDC recommends different diagnostic times. You can test as many insecticides you want. I think here we have around 25 different insecticides. It depends, everything depends on how many larvae or adults we have. So we can test many of them. So when you expose these mosquitoes, you count every 5 minutes what's the proportion of knockdown. So each of these points is like a bottle with 25 mosquitoes, and they show like at minutes 5, there's very few knocked mosquitoes. As time increases, more mosquitoes are knocked, and then you get like your curve. But really what you're counting here is at the end of your diagnostic time. CDC recommends 30 minutes, right? So they say that this is a susceptible colony. So most of your susceptible mosquitoes should be dead by 30 minutes. My susceptible colony didn't die at 30 minutes, right? So, but they died at night at 60 minutes. So those are things that we can adapt to our systems. So here we end up like, this is a susceptible colony. If you were going to test a resistant colony, you would see, for example, that at minute 60, you will end up with only 40% mortality or 0.4 knockdown proportion. So that's where you have to make decisions about what to do next. So for example, if you do the bottle bioassay, these are like the thresholds for insecticide resistance recommended by the CDC. And if you have less than 90% mortality, then they say you have resistance, right? So what you need to do is to try to figure out what's the mechanism, to see what's the intensity, so you can increase the dose of the insecticide and see what's the threshold for these mosquitoes. You can do field testing to see if that resistance really affects your operational control, right? And then you can decide whether to use alternative control methods, or switch from an insecticide, or just stop spraying and do something else, right? So that's this part needs a lot of work, right? So it's very what's the word? The thresholds are very, very abstract, and they're not based in data, right? So they just say this 90%, and that's it, right? We don't know exactly what does that mean on the field. So that's where we need a lot of research. Okay, so the last part is just the case of pyrethroid resistance in in Culex mosquitoes, I was telling you that there's like a lot of pyrethroids are very popular. They're cheap to produce. They have like you can store them for long periods of time. They kill mosquitoes very fast. So people like pyrethroids. And these are sodium channel modulators. So they act at the neuronal system. So if you remember, like there's like the neuron, it has an axon, right? And in the axon, there's where the action potentials go through to communicate from neuron to, well, throughout a neuron to a second neuron. So when this membrane is full of ion channels, sodium channels. So these channels allow this action potential by opening and closing. So when they open, there's like an influx of ion channels inside the neuron, and then that generates an action potential that goes forward. So these pyrethroids, what they do is they bind to these ion channels, and the ion channel has to open and close, open and close. But when the pyrethroid binds, the ion channel cannot close, right? So it's open for longer periods of time. That allows like more ions inside, and that results in depolarization of the membrane, and they stop the action potential, and that results in that the mosquito cannot move anymore, right? So they have a very like tremors, convulsions, incoordination, and knockdown. The mosquito will get knocked very fast. They cannot move, they cannot fly, they will stop breathing and die, right? So that's their mode of action. So this is just a picture of these mosquitoes exposed to a pyrethroid in the bottle. The ones in the front are alive, right? The ones in the back, they are knocked. So that's how a knocked mosquito looks. And they can either recover or not. So it depends on the dose of the insecticide. So the major mechanism of this knockdown resistance is mutations in the sodium channel. This is just like a picture of how sodium channels is a very large protein embedded in the membrane. It has several segments. So each segment forms like is 4 subunits, right? And these 4 subunits mold in a way that they form a pore. And then some regions of the protein form the pore, right? So usually this is S5 and S6 regions of each subunit will form the pore. So they have found that most of the mutations associated with pyrethroid resistance are usually in these areas, right? In this one. There's one mutation called leucine 1014 phenylalanine that you can find in several species of insects that go from cockroaches to houseflies to mosquitoes to Drosophila. So this mutation is being converging in different insect species, which means in resistant mosquitoes, so, resistant insects, which means that this is an important area for the binding of these pyrethroids. So this is just a study in what instead of doing bioassays, they jump directly to go to find the mutation in mosquito populations. This is a study in California. Usually you collect as many mosquitoes you can from an area, and then you do DNA extraction, you do a PCR, and try to find the mutations. In this case, the phenylalanine 1014 mutation is when a mosquito is homozygote for the mutation in red. So if you see there's like a lot of red in this central area of California, in Culex pipiens, lower in the coast, right? But the mutation is there and is very, very well widespread. For Culex tarsalis, there seems to be less mutations, right? But there's some populations that are developing this resistance mechanism in these areas. So what we want is to prevent getting into the Culex pipiens situation, right? We want to keep these mutations at a very low frequency. So this is another example, is Culex quinquefacialis, I cannot say that, in California. So they again collected several mosquitoes. Again, the resistant is in black in these bars in each area collected in 2014. Again, there's a lot of resistance in those. So the idea is we don't know what, because we do the bioassays in the lab, and then we do the molecular bioassays. You say, oh yes, it's resistant. But what does that mean in the field, right? That's the main question. Because sometimes what you find in the lab it doesn't mean anything in the field. So there's different ways of testing these. There are some like semi-field conditions where you have a truck applying the insecticide in the way they usually do, and you set up like these semi-field studies where you put susceptible and resistant mosquitoes, for example, or local mosquitoes and your susceptible colony. They will get those droplets of the insecticide, and then if your local mosquitoes don't die, then you know that they have resistance, right? Always you will know that the insecticide is working by using a susceptible strain. So there are different ways in which you can understand this resistance. This is another one of those studies. They spray different formulations of insecticides. Most of them are like permethrin, pyrethroids, right? And one is malathion. And then they put their mosquitoes, like the colony and the North Shore, and the area that they have. So at 48 hours, most of the mosquitoes are dying, 100% mortality, right? But there are some areas, or for some insecticides, for example, this evergreen, where they only got like 76% mortality. So that raises a concern, right? Not all of the mosquitoes, but again, 76%. I don't know for you what you think, but for me, it's like they're killing still a lot of the population, right? So, but still, we don't know what's enough and what's low, what's much. So there's actually no thresholds to decide what's a failing operational activity. So I think EPA says 80%, but no one really tested them, right? Because they are difficult to test. So, okay, what happens when you find insecticide resistance, right? If you find, oh, there's a lot of resistance with pyrethroids, like, what should we do, right? Definitely not using pyrethroids anymore. But again, you have to, there are different strategies on insecticide resistance management, mostly recommended for agricultural use, where you can switch from one insecticide with one mode of action to another one, right? So you can do rotation. So in a single year, first apply a pyrethroid, then you switch it by a organophosphate, then again a pyrethroid, for example, right? Over time. There's a different method called mixtures where you combine two insecticides with a different mode of action in the same formulation. So you apply it. So the idea is that it's very unlikely that a mosquito will have resistance to both insecticides. So it's very likely that you will kill it. So that's the assumption. And then you have mosaics where you can apply your different molecules of insecticides at the same time in different, especially different, right? So different insecticides at the same time, different spaces. Of course, some of these are very difficult because here it means that your mosquito control program needs to have all of the insecticides at the same time, right? And they have to be very well, like the logistics of knowing, oh, I'm going to apply this in this quadrant, this in this quadrant. So it gets more complicated. But that's what we have as insecticide resistance management. And there are some studies being done in doing this rotation or mixture schemes well, I'm running one in Mexico, for example, just to I tested rotations. that didn't work for what we have. Now we're gonna switch to mixtures because it's we have very strong pyrethroid resistance there. So, okay. So the challenge here is when I'm taking, well, when I'm saying no rotation, but we only have, remember, organophosphates and pyrethroids. So there's not many molecules we can rotate. So also there are no guidelines to when and how do we rotate insecticides based on what, like how we decide to switch from one to another. So that's one of the research that it has to be done. And also we have that several counties don't like to use certain insecticides, which is valid, right? So that leads to options for new molecule developments or just there's a lot of things that we have to figure out here. So with this, I think this is gonna be the last one. So it's like surveillance. Okay, so we have to think more about integrated pest management. We cannot only rely on insecticides. It's better if we use insecticides only for emergencies, right? So that's the idea. But that means that you have to do surveillance to be sure when to use that insecticide. You have to ask people to use personal protection. You have to reduce breeding size when possible and do management of these chemicals if possible. So just to finish, there's a lot to do. There's a lot of research and questions to figure out in this area of insecticides. So I hope you you enjoy the talk, and maybe one or two things are very important from this talk. So let me know if you have any questions, or feel free to. Oh, here's my email. And if you have questions or you're interested in sending samples, you can either write me or write Tim, and we can figure out how to do the logistics of sending mosquitoes or sending insecticides and so on. So is any one of you doing insecticide resistant testing in your areas? No? Well, if you are interested or you know someone that wants, send them to me. Yep. Okay, well, I think I'm gonna finish here. I think Tim is going to send you an email after this. Let me know if you have any questions after this. Just write me an email and we can continue the conversation. Well, thanks for your time, for being here, and have a very nice weekend. Bye.
Thank you. Bye-bye.
Thank you. Bye-bye.